Hi, everybody. In this video, we're going to talk about review part 4, focusing on the cell cycle and mitosis. But before that, we're going to take a look at sexual reproduction versus asexual reproduction. Obviously, bunnies cannot go through asexual reproduction. This is just an analogy. So over here, we can see this parent. For asexual reproduction, we start with one parent. We end with two offspring. They're identical copies of the parent, but really, you just have one cell growing, splitting up. And then you end up with two cells that are identical compared to the original cell that you started with. The original cell is no longer around. Everybody's identical. All the genetic information is identical as well. Versus on the right side, you have to have at least, you have to have two parents and at least one child. And the child is a mixture of the two parents' genetic information. And also, if you look at the offspring, it will look like one parent and also similar to the other parent as well. All right, this right here is a comparison chart between asexual versus sexual reproduction. There's no one strategy that's better than the other one. This is simply how evolution works. Whatever worked out for whatever organism is what works. So take a look at our first row. Asexual reproduction requires one parent. Sexual reproduction requires two parents. What type of organisms go through this asexual reproduction? All the prokaryotes go through asexual reproduction. Some eukaryotes, such as plants and some fungi, go through asexual reproduction as well. Sexual reproduction is for eukaryotes only, but we're not saying that all eukaryotes go through sexual reproduction, but a lot of us do. What do the offspring look like? Asexual reproduction means identical to the parents and to the other offspring, versus sexual reproduction will be a combination of both parents. In terms of genetic information, DNA is identical over here as well. And then on the right side is a combination of the two parents. Advantage of asexual reproduction is that it's fast and it uses less energy. A disadvantage of this is because everybody is the same, all the offspring, the entire little population is identical. That means if there was an environmental change, then it's harder for the population to adapt to the environment because everybody is the same, right? If the environment is perfect, perfect. If the environment is no longer perfect, everybody might not make it. So that's a disadvantage. The sexual reproduction advantage and disadvantage is just the flip of whatever the asexual reproduction has. So the advantage is it has a higher genetic diversity and more adaptive to environmental changes. And the disadvantage is that it's slower and it uses a lot of energy. So you can go ahead and take a look at the answer key over here. Feel free to pause and take notes or whatever you need to do. All right, moving on. We have the next part of our answer key. We have question number 22. I'll just explain it a little bit. For 22, it says, as the cell gets bigger, does it become more efficient or less efficient at transporting nutrients or waste in or out of the cell? So you can think of a big cell as a football stadium, and you can think about a smaller cell as the tennis court, right? You can open up 20 doors for a football stadium and maybe only 5 doors going in and out of the tennis court. It's still going to be much more difficult for people to get in and out of the football stadium because it's a lot of space, right? Even if you open up a lot of doors, it's still a ton of space to fill. Actually, relatively, you have a lot more space to fill than there are doors available for the bigger football stadium. So when a cell gets bigger, the amount of surface, like what's on the outside, in comparison to the volume, if you were to scale it, it actually has less surface. So obviously a bigger, like if you imagine a big ball versus a smaller ball, right? The big ball obviously will have a lot of surface on the outside compared to the smaller ball. However, relative to the size, the big ball actually has less surface. Okay, so why is this a big deal? When a cell gets bigger, it becomes less efficient in terms of transporting nutrients in and letting the waste out. And this is really important because this is why cells are small, one of the reasons, okay? And also, if you think about a mouse versus an elephant, the heart rate and the metabolic rate of a mouse is much higher than that of an elephant. And the reason for that is because when an animal is smaller, it also has higher surface area to volume ratio, which means it loses heat more easily. It has a lot going on in its body. So it needs a lot of heartbeat and a lot of, metabolism, the chemical reactions. Okay, question number 23. Why does DNA need to supercoil into chromosome? I'm going to draw this real quick. DNA normally looks like this. So just a bunch of squiggly circles, okay? And there are multiple DNA in your cell. I'm just going to draw two as an example. So there are two DNA inside the cell. And it's right where the reflection is. So what happens with this squiggly DNA is that it needs to supercoil, which means it will go from this to more like this, and go from that to more like this. So you will wrap it up nicely in all different kinds of ways, and eventually the DNA becomes an X shape. Okay, so I'm drawing a bunch of squiggly, but it's a lot more organized. So this is after DNA replication is already done. When a cell needs to duplicate, the DNA needs to replicate, right? Because you need double the amount of everything. But once you do that, our cell also needs to go through mitosis or meiosis to split up the DNA and everything else inside the cell. And it's really hard for you to split up all these mess, right? It's like putting multiple strings of Christmas lights into one bucket. Next year you take it out, it's going to be impossible to separate them. However, if you wrap it up nicely into just two bundles, it's much easier to separate them apart. So what we see over here is actually a duplicated chromosome. You can look at question number 24. We're talking about the same thing. So a duplicated chromosome has identical DNA on both sides. So what's on the left side is what I have in the box is the same as what's on the right side. They're identical copies of DNA. So this is not asexual reproduction. We're not reproducing yet. It's just replicating the DNA. So now that you have your DNA all organized and you have one side over here, one side over there, it's very easy to then pull the two apart during mitosis. You will call the one side sister chromatid compared to what's on the other side. Okay, overall, it's a duplicated chromosome, and where the two connects, right in the middle over here, is called the centromere. Okay, and the last thing we have, number 25, prokaryotic cell division is asexual reproduction, but specifically is called binary fission. Okay, I'm gonna move on and talk a little bit about the cell cycle. So the cell cycle goes G1, S, G2, and M. Those are 4 phases of the cell cycle, which means if a cell is usually in a cell cycle if it needs to divide. If it does not need to divide, it's in a different phase. But if it needs to divide, you go from 1 cell to 2 cells, whether it's for asexual reproduction or it's just for you to have more cells inside your body, you're growing taller or you're gaining muscles, you're making more cells, right? So how do you make more cells? You have to go through the cell cycle. Okay, so starting with what is a cell cycle? It helps the cell grow and divide. The cell cycle has G1, S, and G2 first. That's called the interphase. Interphase means in between, right? Inter is in between. Interphase is really talking about in between cell division. So if a cell needs to divide, it needs to go the in between cell division and then actually divides. And then it'll go through the in between cell division phase again and then divides if the cell actually needs to divide again. So the interphase helps our cell prepare for cell division, because you can't just divide, you gotta prepare for it, right? Just like your final exam, you gotta prepare for the final exam. You prepare for the exam, and then you take the exam, and then you prepare for another exam, and then you take another exam. It goes on and on. Okay, the preparation phase takes a lot of energy and it takes a lot of time as well. It just makes sense, okay? All right, we're gonna take a look at the overview interphase. We have G1, which really stands for gap 1 phase, because it's a gap, it's in between, right? Gap 1 phase, we have the S phase. S stands for synthesis. Synthesis phase allows DNA replication. Synthesis is really synthesizing DNA. So we turn this one squiggly line of DNA. We make a copy of it, and it really becomes like a bigger bundle of squiggly lines, okay? It doesn't look like this yet. The DNA is not gonna supercoil into this until prophase of mitosis, but to make it just so you can see, right? If we were to bundle up our DNA, it will look like this. Instead of one copy of a DNA, now you have 2. Okay, that's our S phase. We have G2, gap 2 phase, and then M for mitotic phase, mitosis. So mitosis and cytokinesis, and that's where the cell actually divides. And once again, so we have the interphase in between cell division, and then we have the actual cell division for the M phase. All right, moving on. Let's take a look at G1 phase first. GAP1 phase has a lot going on for it. It's the very beginning of preparing yourself for the whole process of cell division. So many things need to happen. Okay, so this is just a summary of some of the main events in G1 phase. So number one, cell increases in size. So apologize for the grammar. There should be an S after increase. So you have a cell, right? You're trying to make two cells out of it. So you need to grow the cell to double the size at some point. It has not happened yet. It's just growing a little bit to make space for more content inside the cell. Number two, right? Why would you need a bigger cell? Well, you need to make some new proteins or organelles. Proteins are in charge of all different kinds of chemical reactions, or maybe they're receiving signals. They're the acceptors of certain cell signals. That we can find proteins in the cell membrane. All of the proteins, you need to start making more of it. Proteins are really, really important. The DNA is also really important inside a cell. But DNA, eventually, a lot of it will take that instruction in the cell and then make protein using that instruction. So you need double the amount of protein. Organelles, right? If you have mitochondria, you need more mitochondria, right? You need more Golgi, you need more rough ER, all of it. Okay. Number three, as like we said, DNA is very important, so we need to prepare for DNA replication, which is our next step, right? It's a big process over here. We need to go from one time the information, double it, right? Every single DNA base, you need double the amount of it. So you need to prepare for DNA replication. So those are some of the main events for G1. All right. Then we're going to take a look at G1 checkpoint. I like to hit this little bell for checkpoint. Okay. So just like airport security, right? You go to the airport, you don't immediately get on the plane. There are different levels of security that you need to get through to make sure that nobody's going to hurt anybody, right? Starting with the G1 checkpoint. The G1 checkpoint, before we go into the next phase, we need to go through this checkpoint. What does it check for? It checks for number one, nutrient. Cell cycle requires a lot of nutrient. You're literally taking one cell and making two cells. And it doesn't seem like a big deal. It happens inside your body all the time, but a cell is very complicated, as you learned previously. So all of the different mechanisms, we need double the amount of it. So we need a lot of nutrients for it, right? That's why you need food. Number one. Number two, it's called growth factors. So a cell receives all kinds of information, all kinds of chemicals that tells it to either divide or not to divide. Growth factors is a type of external regulatory protein. So what it is that when a cell doesn't receive a growth factor from a neighboring cell, it should not divide. If there is an injury or some sort of situation going on inside your body that requires you to make a lot more cells, oftentimes cells will send signals to each other and say, it's time to divide. It's time now, okay? So growth factor is something that allows for the cell to divide. So before the cell goes past the G1 checkpoint, it needs to make sure that it actually needs to divide. And how does it know? It will check for the signal to divide or not to divide. And in this case, it's a signal to divide. If the growth factors are absent, it's not supposed to go past the checkpoint. And if it is present, right, the growth factor is received, and you're fulfilling all the other requirements, then you can go past the checkpoint and go into the next phase. Number three is DNA damage. If the DNA is already damaged, if your DNA is supposed to look like this, right? But then there's actually a break in the middle. Well, that's a problem. You don't want to make more of that, right? If you already have a paper that's written incorrectly and you make 20 copies of it, bad news. We don't do that, right? So what do? If you don't have enough nutrients, or if you don't have the growth factor, you just simply do not need to divide, or you do want to divide, but the DNA is damaged, what do? We try to fix it. And we try to fix it in a phase called G0 phase, which is called the resting phase. As you can see, it's not G1, it's not GAP1, and it's not GAP2. It's a phase that is actually outside of the cell cycle itself. It's like you're going around the roundabout, but you're actually going off the roundabout now, okay? You're no longer in the circle, you're out, okay? And that's not a bad thing. A lot of our cells are in the G0 phase because it simply does not need to divide. A lot of our neurons are in the G0 phase. It's doing its job, it's receiving the nutrients, it's doing whatever it's supposed to do as a cell. It's fulfilling its duties, but it does not need to divide, and that is fine. It will stay in the G0 phase until something happens. Or maybe it just stays in the G0 phase for its whole life. It's fine. Okay. Or a cell needs to divide. Your skin cell needs to divide and make more skin cells. And it's checking for nutrient, growth factor, DNA damage. It's checked for growth factor, but it has not received enough nutrients, or it has not, it does have a DNA damage. So then your cell will take a pause, right? Take a break, take a little nap, figure out what's wrong with it, and try to fix it. So also during this time, the cell is still functioning, it's fine. The cell is not dead. If there is a DNA damage, we'll repair it. Okay, so that's G0 phase. You can go from the G0 phase back into the G1 phase, make sure everything is fine, and then move to the S phase. So now let's move on to the S phase. The S phase is the synthesis phase. We're synthesizing DNA. So DNA replication over here. The event that takes place in the S phase is called DNA replication, obviously, and we're making duplicated chromosomes. The wording is a little confusing over here because the chromosome technically is referring to a duplicated chromosome like this that is already supercoiled. But like I mentioned earlier, the DNA is not supercoiled yet. We've simply made double the amount of it, okay? But I drew it in the X shape so you can see that it is duplicated. But in reality, in your cell, you cannot see the X shape yet. Like it's not supercoiled yet. You just see, it's a dark mess inside the nucleus. Okay, so during the S event, right, if you have 46 chromosomes, 46 DNA, you will still have 46 chromosomes after DNA replication, but you'll have 92 DNA instead. So every single DNA that you have, you will have double of the amount. And the double each duplicate will stick to each other. So that's how you can tell the difference between homologous chromosomes versus sister chromatid. We'll talk about homologous chromosome on the next part of the review, but in terms of sister chromatids, right, how do you tell it's a sister chromatid? You have to have two DNA that are actually attached. If they are directly attached, that's a sister chromatid, but it is still one chromosome, okay? There is no duplicated chromosome if it's just one. Sorry, there's no homologous chromosomes if it's just one chromosome. But one chromosome can have one DNA, or it can have two DNA, two sister chromatids attached to each other, but it's still one unit because they're attached. Okay, so once again, one DNA, two DNA. This is just for every DNA, right? If you have 23 DNA, now you have 46 DNA. But the chromosome number does not change, as you can see. Then we have an X checkpoint. S checkpoint checks for DNA damage again and DNA replication error. The DNA damage, once again, is talking about more like an actual damage, like your DNA is broken. DNA replication error means maybe you're supposed to have ATT, GC, and you're supposed to replicate it to the other strand, TAA, CG, right? But maybe there is a replication error. So instead of this G at the last base, you end up with a C. C is not supposed to pair with C. That's a problem. So your cell will be able to go along the DNA and check for errors. And it checks for the errors, sees that there is a problem, removes the C and adds a G into it. Can you see it? Oh, it's on the other side. Right here. It's right here. It's a little messy. Okay. All right. So that's the S phase. We're moving on to the next one. We have the G2 phase. GAP2 phase. Everything needs to be prepared now. We are going into our actual mitosis very soon. Okay, so everything needs to be prepared. So G2, all the organelles and molecules, your cell needs to be actually almost double the amount of nutrients, the size. You need all the organelles already made. Your DNA is already duplicated, but we still have a checkpoint. This checkpoint checks for cell size. We need a cell big enough before we try to divide it. Otherwise, your cell becomes smaller and smaller and smaller, and then you have no cells left because it's too tiny. Number 2, once again, we check for DNA damage. Again, we always check for DNA damage. Up until this point, we don't check for DNA damage again. Once we pass this checkpoint, and if everything is fulfilled, then we will go to our very last step, which is the mitotic phase. Mitotic phase is not the same as mitosis. However, it does include mitosis followed by something called cytokinesis. So mitosis is the cell trying to divide, but it's not a fully divided yet. Like everything is ready. You have your cake already baked, but you have not cut the cake, okay? Cytokinesis is you taking the cake, everything is ready, it's all decked out, you put the frosting on and everything, and you actually cut it. That's the M phase. M event. What we're really trying to do is to make 2 cells, and we're finally doing it 1 cell division, resulting in 2 daughter cells. We'll talk about the details. But overall, we just want to take every single DNA. We want to split up the 2 sister chromatids, and then this will end up in 1 cell, this will end up in the other cell. And then also every organelle that you have inside the cell, you have duplicates of it, you want to divide up the duplicates as well. We have 1 parent cell, we have 2 daughter cells. There is also an M checkpoint. This checkpoint is really, really, really important, just like all the other ones, but this one's really important as well. Okay, so what is going on over here? We have our 2 copies of our DNA. Let me clean this up a little bit. We have our 2 copies of the DNA, and what we really want to do is to divide up the 2 copies, our 2 sister chromatids. So how would you do that, right? Earlier, you want to untangle your Christmas slide. And if it's already tangled, this is inconvenient. But if it's already bundled up, it's convenient. You just split it apart. Well, how do you split it apart? You have to take one side of your Christmas slide with one hand, the other side, and then you split it, okay? So we have our 1 copy of our DNA. On one side, there's not, the line is just to help you see the 2 sides. And then what happened is inside the cell, we make something called spindle fiber or mitotic spindle. Kind of like a rope that ties onto the 2 sister chromatids, and it's just pulling it apart, okay? So during the mitotic phase, during mitosis, all your chromosomes will line up in one line. So you will have another chromosome, right? Also on the bottom like this. And then you'll line up and get pulled, just like that, okay? And then it'll get pulled apart, the 2 sister chromatids pull apart. Eventually you have 2 cells. So the M checkpoint checks to make sure that the spindle fibers are actually connected, because if you don't have the correct connection over here, for example, then both of these 2 copies of DNA is going to get pulled to the same side. And that is not ideal. You're making 2 cells. You need the right number of DNA in all your cells and not just one of them, okay? Well, also the other cell will have too many. That's also a problem. All right. So that is our entire cell cycle. Let's take a look at the answer key. Here's an image showing you the cell cycle. So how can you tell where we start, right? We have A is the entire thing. Well, 3 slices of the pizza pie. So that has to be your G1, S, and G2, but together A is called interface. B is our first part of the interface, so that's G1. C is S phase because it's the next one. D is the third part of our interface, which is G2. And B, C, D, where's E? Okay. E, as you can see, is splits up into like the K, sorry, K is our mitotic phase, right? But you can see K is split up into many different smaller slices. So what is going on over here? We have this first part, which is mitosis. And then we have what follows mitosis, which is cytokinesis, where the cell actually cut. But within mitosis, we have prophase, metaphase, anaphase, and telophase. So 4 little phases allowing our mitosis to actually happen. So we'll talk more about that in a few minutes. So we have D, which is G2, prophase, metaphase, anaphase, telophase phase. And this whole thing is called mitosis. And this is our mitotic phase. And then we have J, cytokinesis, and the last one, the one goes out of the G1 phase, is our G0 phase. And you can pause and take a look at the answer key as needed. Okay. Whew. Last part of the cell cycle. We're not done. We also have mitosis. You can also pause and take a look at the answer key over here. But overall, we've talked about all the different checkpoints. The last part over here compares a tumor and the repairing of like an injury. So both of those allow cell division. They're both very, they both allow 1 cell to become 2 cells and more cells. You're making more cells. But the mechanism of making these cells and how we get there is very different. When you're repairing an injury, this is supposed to happen. So this is a process that is actually regulated, right? It's you're going through the green light and you're not going through the red light. There is no problems. Versus if you're going through the red light and the green light at the same time, this is going to be a huge mess, right? This is accidents. waiting to happen, and it happens in some people's bodies. So a tumor is unregulated when a cell is not dividing, and when the cell is supposed to form one surface and not just a ball of random cell cluster while it's doing that, even when it's not supposed to. So that's a problem, okay? All right, now our very last part of this lesson, we're gonna take a look at mitosis. Mitosis starts with interphase. Interphase, DNA is unreplicated, so that's why I have wiggly fingers. The nuclear envelope is still intact, as you can see over here. So that's interphase. We have prophase. Prophase, I'm putting together an X shape, but I put it on the side because it's not lined up yet. So all the DNA is condensed. That's why you see the X shape and not the wiggle DNA anymore. So this is our prophase, okay? Nuclear envelope also breaks down because the DNA needs to be able to move around the cell. And with the nuclear envelope, it's trapping the DNA. You can also notice on this picture that we have now these spindle fibers being made by the centrosomes. You don't need to worry about that for our final exam, but still just, general knowledge, hear about it. Spindle fibers reaching out to find our DNAs, okay? And they are going to then connect to these duplicated chromosomes, duplicated DNA, duplicated chromosomes. Okay, then we have metaphase. So I'm putting the X shape in the middle and I'm moving it up and down because I don't have 8,000 fingers. So there's an X shape over here and one over here, one over here, one over here. They're all in the middle, okay? On the screen is one line horizontally. It doesn't matter. You can take the cell, flip it, okay? But I like to do just up and down is fine. So what's happening over here is that we say sister chromatids line up at the metaphase plate, which means they are chromosomes lining up, but really we have the sister chromatid, one on each side, one on this side, and one on this side, right? So they are lining up. So then the sister chromatids can be pulled apart during anaphase. You can also help yourself remember metaphase, M for middle. They're lining up in the middle. And then we have our anaphase. A for away, they're moving away from each other. And who's moving away from each other? The sister chromatid, if they are connected correctly to the spindle fibers previously. Anaphase sister chromatids pull apart, and then we have telophase, DNA uncondensed, nuclear envelope reform, and cytokinesis where the cell actually gets cut in half. So how can you tell there are different phases, right? How can you tell which phase is which? We have our prophase, right? If you see nuclear envelope breaking down, but there's only one of them, that's prophase, because you can't tell if it's breaking or making. But if you see two of these circles, that has to be telophase. If they're lining up in the middle, horizontally, vertically, doesn't matter, middle, metaphase. Anaphase, sister chromatids being pulled apart. We no longer have the duplicated chromosome. We no longer have the X shape. We see individual lines. The little lines are being pulled apart. That's anaphase. And lastly, cytokinesis, you have solid circles of the nucleus and the DNA are inside, but they're now no longer the X shape. They also might not be the X shape over here. As you can see, they're detangling, decondensing. There are two types of cytokinesis. One is for animal cells, cleavage furrow. Essentially, it's like you have a Play-Doh, and then you squeeze it right in the middle, and then it gets split into two halves. That's for animal cells. And plant cells, it's a cell plate. Cell plate is a growing cell wall. Cell wall is very rigid. You have to actually build it. Okay. And once again, as you can see, mitosis is prophase, metaphase, anaphase, and telophase. But mitotic phase really is mitosis followed by cytokinesis. That's why I put the C right here in a parentheses. Okay, you can take a pause and take a look at the different phases and the events that we just talked about. And a quick comparison between mitosis and meiosis. Or let's just take a look at mitosis first. Mitosis goes from one cell to two cells. The daughter cells are identical to each other, and they're identical to the parent. You only split up one time. You start with a diploid cell, you end with a diploid cell. Or you start with a haploid cell, you end with a haploid cell. We'll talk more about diploid versus haploid during the next review, but it's just whatever cell you start with. The number of chromosomes stays the same. If it's 46, it's 46 now. If it's 20, it's 20 now. If it's 30, it's 30 now. It doesn't matter. That's mitosis. And I think that's it. And then we will look at meiosis. In comparison, we have one cell splitting once, splitting again, right? You might say, oh, it's splitting here again. Well, there are two like each cell really only gets split twice. We get one cell split once, right? And then they each split again. So it's really meiosis just splitting 2 times. We have 4 cells that you end up with, and they're all different from each other, and they're different from the parent as well. Also, after the first split, these 2 cells are supposed to be different. Okay. So another comparison, we have mitosis. It's all sister chromatins lineup. It's a little hard to see, but it's all one file line versus meiosis has sister chromatins on this side and sister chromatins on this side. So we're really splitting up the homologous chromosomes. Okay, here's the comparison chart. 1 cell, 2 cell, what? 1 parent cell, 1 parent cell, that's fine. 2 cells as a daughter cell, 4 daughter cells, diploid, diploid, usually diploid, and then 2 haploid. We cut the chromosome number by half. Somatic cells are cells inside our bodies for a multicellular organism. Our lung cells, our blood cells, our neurons, those are all somatic cells, not the gametes, not the sperms and eggs. So mitosis helps us produce somatic cells for growth of our bodies and for repairing of injuries, versus meiosis is really for reproduction of organisms that go through sexual reproduction. Are daughter cells identical? Yes, they're different. Number of cell division 1 versus 2, the amount of genetic diversity low because everybody's the same. Meiosis produces 4 cells, and all 4 cells are different from each other. What's lining up? Sister chromatid line up at the metaphase plate during mitosis versus meiosis is homologous chromosome during meiosis 1 and sister chromatids during meiosis 2. And that's it. All right. That's the end of the video. Come back for part 5 to learn about meiosis. Goodbye. Good luck.